Welcome to the deep dive. Today, we're really immersing ourselves in the story of the 1918 Great Influenza. I mean, a cataclysm that struck a world already deep in the chaos of total war. We got a pile of sources here looking at the science, the societal breakdown, and that frantic race for a cure.
And we really have to start with a scale.
Yeah.
It's almost unbelievable. The lowest estimates back then, 21 million deaths globally.
Which is already staggering.
Right. But modern epidemiologists, looking at the real data now, they estimate it's actually closer to 50 million, maybe even possibly up to 100 million deaths.
100 million. In a world with, what, less than a third of the people we have today, it's just immense.
Yeah.
But the thing that really chills you, the thing that makes 1918 stand out is who is dying.
Exactly. Normally, flu.
Yeah.
It's tough, but it follows a pattern. It hits the very young, infants, and the elderly.
Yeah.
Their immune systems are either not ready or worn down.
Yeah, predictable, almost.
But 1918, roughly half of everyone who died were young adults, people in their 20s and 30s. The absolute prime of their lives, peak physical health.
That's just terrifyingly backwards. No wonder Surgeon Harvey Cushing, who saw it firsthand, actually got sick himself, he called them doubly dead in that they died so young.
It's profound.
So, okay, before we even get to the virus itself, we need to look at the medical world it crashed into, because this was right in the middle of a huge scientific revolution in medicine.
It absolutely was.
Yeah.
If you just went back, say, 50 years before 1918, medicine hadn't really changed much conceptually since Hippocrates. That's
Right.
over 2,000 years.
Wow.
You had figures like Benjamin Rush, signer of the Declaration of Independence, highly respected. But his theories, still based purely on logic, observation, he thought fevers were caused by convulsive action in the capillaries.
Sounds smart, but not really testable, is it?
Not at all. And there was this real intellectual arrogance back then, too. Physicians looked down on surgery. They saw it as just a mechanical skill, something for barbers, not for proper doctors.
That's beneath them.
Totally. The big change, the fundamental shift, happened when doctors started asking a different question. Not just, what can I know? But, okay, how can I actually know this is true? That's the leap.
The scientific method taking root. You get Francis Bacon challenging Aristotle's deductive logic, saying it's useless for finding new truths about the physical world.
Exactly. And then you start seeing the proof in the pudding. William Harvey tracing blood circulation, that was way back in 1628. James Lind, 1753, runs the first controlled experiment, shows limes prevent scurvy. Simple, but revolutionary.
And surgery starts getting respect.
Yeah, thanks to people like John Hunter.
Yeah.
He really pushed for rigorous study, elevating it beyond just, cutting. But America was lagging behind for a long time. Europe, especially Germany, that was where the real cutting-edge medical science was happening.
Until Johns Hopkins. That seems to be the turning point for the U.S., just in time for the war. The medical school opening in 1893.
That's the catalyst, absolutely. The Quakers who founded Hopkins, they didn't copy Harvard or Yale. They looked straight to the top German universities. They demanded science, real lab work, rigorous training. It wasn't just about reading books anymore.
And that really brought American medicine up to speed.
It was probably the single biggest factor. By World War I, American medicine was finally catching up, largely thanks to Hopkins.
And there's a key figure in all this, right? William Henry Welch.
Oh, absolutely. Welch is central. His trip to Europe back in 1876, even before the Hopkins Medical School was properly established, people call that trip the most important ever taken by an American doctor.
Why was he so influential?
He was more than just a scientist. He was kind of an impresario. He knew how to direct money, how to spot talent, how to build institutions. He could make or break careers, but apparently he kept his cool, kept perspective. Younger scientists called him Popsy.
Popsy, okay.
Yeah, he had this huge influence, but didn't seem to let it go to his head. He basically built the scientific establishment that was about to face this unprecedented plague.
So the scientific tools were getting sharper, but when the influenza hit, they didn't even really know what the enemy was. It was an RNA virus. We know that now.
Right, and that RNA structure is key. Unlike stable, double-stranded DNA, RNA is simpler, single-stranded, and it's much less stable. It doesn't have good proofreading tools when it copies itself.
Meaning lots of mistakes.
Lots of mutations.
Yeah.
Rapidly. So the flu virus isn't just one thing. It's constantly changing, existing as this mutant swarm. Scientists call it a quasi-species.
Which explains why it's so unpredictable, why we need new flu shots every year. Okay, so let's zoom in. The virus gets into your body. What's the attack plan?
It uses these little spikes on its surface, like microscopic grappling hooks. They're called hemagglutinin spikes. That's the H in H1N1, for
Oh,
example.
okay.
These H spikes fit perfectly onto specific receptors on your respiratory cells, the sialic acid receptors. It's like a key in a lock, or maybe a hand going into a glove is a good analogy. Once it latches on, the virus gets pulled inside the cell, sheds its coating, and its genes basically hijack the cell's machinery.
Takes over the factory.
Completely. Tells the cell, stop doing your job. Start making copies of me.
And then it needs to get all those new copies out to infect more cells. That's the other spike, the N.
Exactly. Neuraminidase, that's the N in H1N1. These spikes are the escape artists. They act like scissors, cutting the connections to those sialic acid receptors on the outside of the dying cell.
Ah, so they don't get stuck.
Right. Without neuraminidase, the new virus swarm, and we're talking maybe 100,000 to 1,000,000 new viruses produced in just 10 hours or so. They'd be trapped on the surface of the dead cell, like flies on flypaper. Neuraminidase snips them free so they can go invade neighboring cells.
Okay, that's the mechanism. But the symptoms were baffling doctors, like Paul Lewis back then. People collapsing looking like they'd been gassed or had pneumonic plague. What was actually killing them so fast?
The autopsies were just grim. They called it fulminating pneumonia. The lungs were wet, heavy, filled with blood, hemorrhagic. Death could come in 24, maybe 48 hours.
So,
And victims turned blue, sometimes almost black. That cyanosis, severe lack of oxygen. But here's the truly awful part, the biological twist.
Yeah.
It wasn't always just the virus directly killing cells that caused the worst damage, especially in those young, healthy adults.
the ones who died most often.
Yes, it was often their own immune system's incredibly strong reaction that killed them.
Wait, what? Their immune response was too good? How does that work?
It's like friendly fire on a massive scale. When their strong immune systems detected this new, aggressive virus, they sounded the alarm. But the alarm was too loud. They released this overwhelming flood of signaling molecules, cytokines, to rally the troops.
Okay, calling for backup.
But it was a tidal wave of backup.
Uh-huh.
This cytokine storm caused massive inflammation, far too much for the delicate tissues of the lungs. The immune system, in its attempt to destroy the virus, ended up destroying the lung tissue itself.
So the body's defense mechanism became the killer.
Essentially, yes. The lungs would fill with fluid and blood from the damage. That's the hemorrhagic pneumonia. We now call it ARDS, acute respiratory distress syndrome. The fiercest immune systems provoked the deadliest response. A brutal irony.
Unbelievable. And where did this monster likely start? The evidence points towards Kansas.
Yeah, the strongest candidate seems to be Haskell County, Kansas. Rural place. There was an unusually severe flu outbreak reported there in February 1918. Local doctor noticed it was different, hitting healthy young people hard.
And from there,
It likely spread to nearby Camp Funston, which was a huge, massively overcrowded Army training camp. Perfect conditions, men packed together, sleeping in shifts sometimes, coming from all over, then shipping out.
so the war machine itself became the distribution network.
Precisely. Troop ships moving soldiers across the country, across the Atlantic. They became floating incubators, spreading the virus globally with incredible speed.
Which brings us straight to the social dimension. This disaster didn't happen in a vacuum. It hit during World War I and the government's priorities. Well, they weren't always public health.
Not at all. The government, particularly the Wilson administration, was focused on total war. And a huge part of that was maintaining morale, keeping spirits high, projecting strength.
So they controlled the message.
Absolutely. You had the Committee on Public Inforgeny, the CPI, run by George Creel. Their explicit job was to forge one white-hot mass of patriotic unity, which meant suppressing any bad news that might dampen enthusiasm for the war.
Including news about a deadly plague sweeping the country.
Especially that. Newspapers widely self-censored or were pressured to downplay it. And this was happening in an already incredibly repressive atmosphere.
You mean the Espionage and Sedition Acts?
Yes. Those laws made it illegal to say almost anything critical of the government, the flag, the military uniform, even if what you said was completely true.
That's terrifying.
People were jailed for criticizing war bonds, for questioning the draft. Famously, they tried to force people to call sauerkraut Liberty cabbage. Criticizing that could get you reported. And you had vigilante groups like the American Protective League, 200,000 members spying on their neighbors, reporting disloyalty.
Imagine living like that. Constant fear, zero trust, and then this incredibly lethal disease arrives.
And hits a healthcare system already stripped bare. The military needed doctors and nurses, desperately. They pulled something like 38,000 physicians into the service, maybe half of all the younger, fitter doctors in the country.
Leaving who behind for civilians?
Often the older doctors, maybe less up to date, trained before the big scientific shifts, or doctors considered less competent.
Mhm.
The civilian population faced a massive shortage right when they needed help most. Plus, cities and camps were dangerously overcrowded due to the war effort. Camp Funston had men sleeping head to toe.
It's a perfect storm. And Philadelphia. That seems to be the textbook example of how badly things could go wrong.
Philadelphia is the nightmare scenario. Experts like Paul Lewis were there. They saw it coming from outbreaks elsewhere. They warned officials. They begged them to cancel a huge Liberty Loan parade planned for late September.
But they went ahead anyway, for
They
morale.
went ahead. The city's public health director, Vilmer Cruzen, kept insisting it was just old-fashioned influenza or grippe. He actively lied to the public. Thousands gathered for the parade.
And the consequence.
Immediate and catastrophic. Within days, the city was overwhelmed. Hospitals filled, then overflowed. Dr. Stengel, a top infectious disease guy there, basically threw up his hands saying, "This doesn't work. That doesn't work. Nothing works."
Just complete helplessness.
It was medieval. Morgues couldn't handle the bodies. They were stacking them up in hallways from floor to ceiling like cordwood, according to reports. Eventually, the city brought in steam shovels to dig mass graves. People just got used to seeing the black mourning crepe on house after house.
And all the while, the newspapers are downplaying it, officials are lying. That disconnect must have created sheer terror.
Absolutely. Because what you read had zero connection to the horror you were seeing, touching, smelling every day. Your neighbors dying, bodies in the streets. That failure of trust, the gap between official lies and reality, that created a unique kind of terror.
Made it worse than just the disease itself.
Much worse. Victor Vaughan, a really respected, sober figure in public health, he warned that this fear, this breakdown of trust, threatened to break the society apart. He actually said civilization could have disappeared within a few more weeks. That's how close he thought things were to total collapse.
Fueled by lies. Okay, so in the midst of this chaos, the scientists, they're scrambling. People like Paul Lewis, William Park in New York, they know they're racing the clock.
They knew they had to make educated guesses, and fast. They didn't have antiviral drugs, obviously. They couldn't target the influenza virus itself effectively yet.
So what could they do?
Their main hope was to fight the second wave of attack bacterial pneumonias that often followed the initial viral infection. Those are frequently the actual killers.
Ah, the opportunistic invaders.
Exactly. And this is where the Rockefeller Institute team, led by Rufus Cole and a brilliant young researcher named Oswald Avery, came in. They'd already developed serums against some of the most common types of pneumococcus bacteria, types I and II.
So they had a potential weapon against the follow-on infections.
They did. It wasn't a cure for the flu, but it could potentially save people from the pneumonia that killed them. So they rushed to mass-produce these serums.
But doing precise science in that environment must have been incredibly difficult.
Oh, completely chaotic. There's a great story about Avery working at Camp Devens, another badly hit Army camp. He was doing critical tests, gram stains, to identify bacteria, but his results were all over the place. Made no sense.
What was going wrong?
He finally figured it out. Desperate, sick soldiers had been sneaking into the lab, drinking the pure alcohol needed for the staining procedure, and then topping the bottles back up with water.
Oh my God. You're trying to save lives with cutting-edge science, and people are drinking your lab supplies.
It perfectly captures the desperation and the sheer mayhem they were working within. Precision was almost impossible sometimes.
And Paul Lewis in Philadelphia, what was his approach?
Lewis was trying what they called a shotgun approach with vaccines. He couldn't target the virus, so he made a vaccine combining killed samples of several different bacteria he suspected were involved as secondary invaders, B, influenza, which they initially thought caused the flu, different pneumococcus types, streptococcus.
Throwing everything at the wall, hoping something sticks.
Pretty much. Trying to give the immune system some kind of head start against the likely bacterial culprits, even if they couldn't stop the initial viral assault.
Did it work?
Not definitively, not in 1918. But Lewis's work wasn't a dead end. His protege, the guy named Richard Shope, built on it years later. Shope was the one who definitively proved that swine flu, which we now know is a direct descendant of the 1918 virus, was indeed caused by a virus.
Ah, closing the loop.
Right. Though Shope also confirmed that the bacterium Lewis was targeting, B. influenza, was a crucial secondary invader that made the illness much worse. So Lewis was on the right track in a way.
And Lewis himself, what happened to him?
A tragic story, actually. He dedicated his life to fighting infectious diseases. Years later, he was in Brazil researching yellow fever, another deadly virus, and he died. likely from a lab accident, maybe contaminated a cigarette. A researcher consumed by the very things he chased.
Wow. So let's try to pull this all together. Looking back with the benefit of all these sources, what's the big picture of 1918?
What was really the first major collision between modern science-based medicine and a completely novel, incredibly virulent pathogen. The science was finally getting somewhere, developing tools, understanding methods.
But it wasn't quite ready for this.
Not even close. And the impact was just massively amplified by the specific circumstances of the war, the troop movements, the government censorship, the breakdown of trust, the shortages. The social context turned a biological disaster into a societal catastrophe.
And the end of the pandemic? Was it the science that beat it?
Mostly. No. It was mainly the virus itself changing. There's a concept called reversion to the mean. The 1918 virus was at an extreme peak of deadliness. Statistically, future mutations were much more likely to make it less lethal, not more.
So it just burned itself out, essentially mutated into milder form.
Largely, yes. We see evidence for that. Cities that got hit later in the pandemic, like San Antonio, they still had terrible death rates, maybe double a normal flu season, but not the explosive, catastrophic rates seen earlier in places like Philadelphia or Boston. The virus seems to have become less virulent over time.
So nature, more than medicine, ended the worst of it.
In 1918, yes. And we shouldn't forget the long shadow it cast. Survivors often had lingering issues. Harvey Cushing, the surgeon we mentioned, he never felt he fully recovered his energy, had neurological problems.
Yes, President Wilson.
That's a huge point historians raise. Wilson got sick during the crucial Paris Peace Conference negotiations after the war. Many believe the lingering effects of the influenza may be neurological, maybe just profound fatigue and what some called a loss of resiliency, significantly hampered his ability to negotiate effectively, potentially changing the course of the 20th century.
That's a staggering thought. A virus shaping global politics, which brings us right up to today, doesn't it? The intersection of this kind of biological threat and our scientific capabilities.
It really does, because scientists now have the tools through genetic engineering and synthesis to potentially recreate the 1918 virus, or viruses like it. They might be very close to figuring out the exact genetic sequences that made it so uniquely deadly, especially to young adults.
And that knowledge. It's powerful, but also dangerous.
Extremely. Which leads to a major ethical dilemma drawn straight from understanding this history. If scientists do pinpoint that lethality mechanism, what should they do?
Publish it. Share the knowledge so other scientists can develop defenses, vaccines, antivirals, specifically targeting that mechanism.
That's the argument for openness. Prepare ourselves for the next natural pandemic, block the mechanism if it reappears.
But the flip side, publishing that information is essentially giving a blueprint to anyone, terrorists, rogue states who might want to weaponize it, creating an artificial plague potentially even worse than 1918.
Exactly. It's been compared to publishing the plans for a biological weapon that could cause a worldwide nuclear-level catastrophe. Do you risk that for the sake of scientific progress and potential future defense?
Or do you keep it secret, potentially leaving us vulnerable if nature throws that same deadly curveball at us again?
There's no easy answer. It's a question that pits the core principles of open science against the potential for unimaginable harm.
A really sobering thought to end on. Weighing knowledge against security in the face of these microscopic threats. A question stemming directly from the lessons of 1918. That's all the time we have for this deep dive.